this video lecture we will be discussing about the Krebs cycle. As we know that we have the biomolecule, that is carbohydrate, protein and lipids. So these molecule will give us energy. Now this energy, how this biomolecule it is able to provide energy? Now these biomolecules are actually break down into small products, that is of glucose, amino acids and fatty acids. So these are finally converted into the acetyl coenzyme A. Now this acetyl coenzyme A, it is finally oxidized through the cyclic process, and at the result of this cyclic process these biomolecule it is able to provide as energy. Now this through cyclic process, when this biomolecule are oxidized, now this cyclic process it is called as Krebs cycles. Now we will be talking about here what the Krebs cycle is. So actually the complete oxidation of acetyl coenzyme A to the carbon dioxide and water and the presence of oxygen, this is called as Krebs cycle. So finally this acetyl coenzyme A, it may become from the carbohydrates, it may become from the protein, it may become from the lipids. So from the all these biomolecule, the acetyl coenzyme A, it is finally we will get from the breakdown of the biomolecules. Now here the oxidation of this acetyl coenzyme A occur and will finally convert it into the carbon dioxide and will form ATP and water molecule. So this overall process occur in the presence of oxygen. Now this process it is called as Krebs cycles. Now you should have to remember that the Krebs cycle it will occur only in the presence of oxygen. Without oxygen there is no Krebs cycles. Now second definition, that is the oxidation of activated pyruvate. So this means that activated pyruvate it mainly means that the acetyl coenzyme A. So what happened, and this the oxidation of the acetyl coenzyme A occur to the carbon dioxide is an aerobic process, means it occur in the presence of oxygen and is carried out by a cyclic metabolic pathway called as Krebs cycle. Now you can see here the oxidation of the acetyl coenzyme A to the carbon dioxide. This occur in the presence of oxygen. First point. The second point, that this is occur in a cyclic metabolic pathway. Now this cyclic metabolic pathway through which the acetyl coenzyme A it is converted into the carbon dioxide and water molecule along with the production of the ATPs. Now this cyclic metabolic pathway it is known as Krebs cycles. Now here are the some of the points related mainly to the Krebs cycle. So the first point, that is about 65 to 70% of the ATP is synthesized in Krebs cycle. As we know that the ATP are mainly the energy currency, they will give us energy. Now about the 65 to 70% of the ATP to provided to our body, that is given by the Krebs cycles. Second point, that is it is the final oxidative pathway for the amino acids, fat and carbohydrates. So mainly when these carbohydrate, fats and amino acid, they are finally oxidation of these carbohydrate, fat and amino acid occur through the Krebs cycles. So third one, it is not only supply energy but also provide many intermediate required for the synthesis of glucose, amino acid and heme, etc. As we have talked about this Krebs cycle, it will give us 65 to 70% of the energy, but only the production of energy it is not the purpose of the Krebs cycle. It will also provide some intermediate compounds. These intermediate compound it will able to convert into many compound. The intermediate it will use for the synthesis or the formation of glucose. It may form amino acid, it may also form heme, etc. So the metabolic pathway it is also used for the synthesis of some other compounds. Now we will discuss here why the Krebs cycle it is called as Krebs cycle. So you should have to remember that the Krebs it will be written without apostrophe. Now this cycle for the first time in 1937 it was described by the Hans Adolf Krebs when he studied on the breast muscle of the pigeon. So on this study he will describe this whole cycle. So that's why this cycle, on the basis of the honor, this cycle it is known as Krebs cycles. So for this discovery, he will get the Nobel Prize in 1953 in the physiology and medicines. Now we will discuss that this Krebs cycle it is also known as citric acid cycles. It is also known as tricarboxylic acid cycles. Now we will look here why this cycle it is called as citric acid cycles. The first name, as this cycle, the first product that is formed in this cycle it is actually the citrates. So that's why this cycle it is also known as citric acid cycles. Now look, you see here the other name, that is tricarboxylic acid cycles. Now why this Krebs cycle it is called as tricarboxylic acid cycle? Because in this cycle we will have three intermediate, that is citrate, isocitrate and aconitate. So these are actually the three intermediate of the Krebs cycle. Now if we look to the structure of these three intermediate of the Krebs cycle, we will see here there are three carboxylic acid group is present in each of the intermediates. So that's why this cycle it is called as tricarboxylic acid cycle. So this is the structure of the citrate. So you can see here these are actually the carboxylic acid groups. So each of the intermediate it will possess three carboxylic acid groups. So this is the structure of the isocitrate. It also possesses the three carboxylic acid group, while the aconitate it also possesses the three carboxylic acid group. So that's why this cycle it is known as tricarboxylic acid cycles. Now look, it see here we will talk here the where the Krebs cycle it is occurs. Now if we talk about the location of the Krebs cycle, so it occur in the prokaryotes as well as in the case of the eukaryotes, but only condition that is necessary for the Krebs cycle that is actually the presence of oxygen. Krebs cycle operate only in the presence of oxygen. If we talk about the eukaryotes, so this Krebs cycle it will occur in the mitochondrial matrix, but in case of the prokaryotes, this Krebs cycle it will occur in the structure that is mainly formed from the invagination of the plasma membrane. Now this structure it is called as mesosome, while mainly in prokaryote the Krebs cycle or it's occur in the mesosome. Now if we will discuss here the enzyme that is mainly responsible for the Krebs cycle, as it contains many of the intermediates. It contains various reactions. So these reactions that are mainly performed by the enzyme. Without enzyme there is no reaction occurs. Now the enzyme that is mainly responsible for the Krebs cycles. So these enzymes that are mainly located in the mitochondrial matrix, because this Krebs cycle it will occur only in the mitochondrial matrix, while the enzyme it is responsible for the Krebs cycle it also occur in the mitochondrial matrix, except one enzyme, that is succinate dehydrogenase, which is present in the inner mitochondrial membrane. So this is actually one of the exception regarding to the Krebs cycle enzymes. Now we will discuss here what are the features or characteristics of the Krebs cycles. Now look at see here the first characteristics of the Krebs cycle. So it is the main catabolic pathway by which fatty acid, amino acid and carbohydrate are oxidized to carbon dioxide. As we have already discussed in the definition of the Krebs cycle, so mainly here the final oxidation of the fatty acids, amino acid and carbohydrate, these are finally oxidized through Krebs cycle, and we will get it the result, that is carbon dioxide and water molecule along with the production of the ATPs. So this is the first characteristics of the Krebs cycle. Now look at see here the second characteristics of the Krebs cycles. Now this Krebs cycle it's occur in the presence of oxygens. This cycle mainly occur in the presence of oxygen. Without oxygen there is no concept of the Krebs cycles. Now the third characteristics of the Krebs cycle, that it also serves as anabolic role and supply carbon skeleton for the biosynthesis of variety of organic molecules or compounds. Now as we have talked about in the first characteristic, that it will play a catabolic role in which the amino acids, fatty acid and carbohydrate that are oxidized to the carbon dioxide. But it also acts as anabolic role, mean it will also synthesize some other organic compounds. So how these organic compounds that are formed? Now we will take here one of the example, that is one of the intermediate of the Krebs cycle, that is oxaloacetates. Now what happens? So this is actually the another amino acid, one of the amino acid among the 20 standard amino acids. Now this oxaloacetate, as we have talked here, that it will provide the carbon skeleton for the biosynthesis of the variety of organic compound or molecule. Now this is actually the keto acids. So it will provide it a carbon skeleton to this. So these are actually the carbon skeleton of the intermediate of the Krebs cycle, that is oxaloacetate. When this carbon skeleton, it will provide to this amino group, or when this carbon skeleton, it will combine with the amino group, or you can simply say that. So when this amino group, it is transferred into the carbon skeleton of the oxaloacetate, it will convert it into the new amino acid, that is aspartate. Now this process, it is called as transamination. Through transamination, the intermediate of the Krebs cycle, it will convert into the new organic substance. In this case, here the example is, that is amino acid that is mainly from the intermediate of the Krebs cycle. So it is actually the anabolic role of the Krebs cycle. Now look at see here the other examples. So the one of the intermediate, that is succinate and succinyl coenzyme A, these are actually the intermediate of the Krebs cycles. So it will provide the carbon skeleton for the synthesis of hemoglobins, myoglobins and cytochrome. So these compounds that is mainly formed from the intermediate of the Krebs cycle. So these examples show that the Krebs cycle, it will also act as anabolic. It will also act the role of anabolism. So you can see here, so this is actually the role of the Krebs cycle intermediates. Now the last characteristics of the Krebs cycle, that is it generates reduced coenzyme, that is NADH2, FADH2, which provide the ATP through electron transport chain. Now these reduced enzyme, that is mainly from through the Krebs cycle, we will get the reduced coenzyme, that is NADH2 and FADH2. So these are actually from through the Krebs cycle. So when these Fa reduced coenzyme, it will pass from the electron transport chain. So at the result, it will provide the ATP. So from the one molecule of the NADH2, it will give us 3 ATP, or you can say 2.5 ATP. Actual result, that is 2. It will give us 2.5 ATP. As the FAD, it will also provide the 2 ATP, or you can actually say that 1.5 ATP. So this is actually the sum of the characteristics of the Krebs cycles. Now we will learn the overall Krebs cycle. So this Krebs cycle, it will mainly start from the initial substrate for this TCA cycle, that is acetyl coenzyme A. Now this acetyl coenzyme A, it will mainly form from the pyruvate by the oxidative decarboxylation reaction. Now this acetyl coenzyme A, after the oxidative decarboxylation of the pyruvate, it will form. Now this acetyl coenzyme A, it is mainly a 2 carbon compounds. So this acetyl coenzyme A, it will combine with one of the substance that is mainly present in the mitochondrial matrix, that is mainly a 4 carbon compound, that is oxaloacetates. So this acetyl coenzyme A, it will combine with the oxaloacetate. So this reaction mainly catalyzed by enzyme, that is citrate synthase enzyme. So at the result, we will form a 6 carbon compound, that is called a citrate. Now this citrate, it is actually the first product of the Krebs cycle or the TCA cycle. So on this basis, the Krebs cycle, it is also called as citric acid cycles. Now what happened here is, so here the water molecule, it is added. Now this water molecule, it will break the bond between the acetyl and coenzyme A. So after the breakage of this bond, energy is released. Now the energy release, it is used for the citrate condensation. So here one water molecule, it is added, while the coenzyme A, it is released here. So after this, the citrate, it will convert it into the isocitrate through unstable intermediate acid it is formed here. So this unstable intermediate acid it is called as cis aconitate. It is actually an unstable acid formed during the conversion of citrate into the isocitrates. So here this reaction, this conversion of citrate into the isocitrate, it will occur in two phases. So in the first phase, actually the water molecule it will remove from the citrate and will form an unstable compound. This is called as cis aconitate. So this citrate it is mainly a 6 carbon compound, while the cis aconitate it will also a 6 carbon compound. This conversion of the citrate into the cis aconitate will occur with the help of aconitase enzyme. Now in the second phase, this the same enzyme aconitase, it will add water molecule to this cis aconitate and will convert this cis aconitate into the isocitrate. So here too this reaction, the conversion of citrate into isocitrate, it will occur in two steps. And the first step, aconitase enzyme, it will remove water molecule from the citrate and form cis aconitate. And the second step, the same enzyme aconitase, it will add water molecule and will convert cis aconitate into the isocitrates. Now and this step, after this the isocitrate it will convert it into the alpha ketoglutarate. So here the oxidative decarboxylation of the isocitrate occur at the result alpha ketoglutarate it will form. Now again this reaction it will occur in two phases. So in the first phase, isocitrate dehydrogenase enzyme it will act on the isocitrate. Here two molecule or two hydrogen ion it is released here from the isocitrate. Now this hydrogen atom it will gain by the oxidized NAD and will convert into NADH2. So here first reduce coenzyme it is formed in these reactions. So when after the removal of two hydrogen at the result NADH and H it will formed here, the reduced coenzyme it is formed here. So actually this isocitrate it will convert into oxalosuccinate along. So these all of these compound that are actually sx carbon compounds. Now here the oxidation occur, mean removal of hydrogen occur here. Now in the next step, the same enzyme isocitrate dehydrogenase it will act on the oxalosuccinates. So here normally the decarboxylation occurs. So in the first step oxidation occur, while the second step the decarboxylation occurs. So this decarboxylation it will occur with the help of isocitrate dehydrogenase enzyme. So it will remove carbon dioxide from the oxalosuccinate. So here the 6 carbon compound it will convert it into 5 carbon compound, that is alpha ketoglutarate. So there is the first carbon dioxide it will remove here. Now look at see here what happened to the alpha ketoglutarate. So again alpha ketoglutarate it will convert it into succinyl coenzyme, another intermediate of the TCA cycles. So simply you have to remember this intermediate of the T cycle, that is alpha ketoglutarate. It is only the one compound or one intermediate, the only intermediate of the TCA cycle that have a 5 carbon. So 5 carbon compound that is present in the TCA cycle, that is actually the alpha ketoglutarate. So again like the previous step, here both oxidative decarboxylation reaction occur. Through this oxidative decarboxylation reaction, alpha ketoglutarate it will convert it into succinyl coenzyme A as occur in these steps. So in this step actually first two hydrogen atoms they are released. So simply the removal of hydrogen it will mainly called as oxidation. So this hydrogen it will form, it will gain by the oxidized NAD. So it will convert NAD into reduced form, that is NADH2. So here second NADH2 it will form in the TCA cycles. So here oxidation occurs. So here acetyl coenzyme A it will add it in these steps. So along with the oxidation here at the same time carboxylation occurs. So second carbon dioxide it will remove here. So this reaction mainly catalyzed by enzyme that is called as alpha ketoglutarate dehydrogenase enzyme. So here this conversion of alpha ketoglutarate into succinyl coenzyme A, it is similar to the conversion of pyruvate into the acetyl coenzyme A, that is mainly the link reaction. So this reaction it was mainly similar to the link reactions. Now what happened to the succinyl coenzyme A? So simply this is actually the 5 carbon compound. So one carbon it will remove in the form of carbon dioxide. So mainly the 4 carbon compound succinyl coenzyme A, it will actually the 4 carbon compound. Now what happened to the succinyl coenzyme A? So simply the what happened to the succinyl coenzyme A? Actually the cleavage of succinyl coenzyme A occurs. So here mainly this succinyl coenzyme A, it will convert it into the succinate. So here the energy is released, and this energy in actually this conversion of succinyl coenzyme A into the succinate, it will occur with the help of succinate thiokinase enzyme. So here GTP it will formed here, and again coenzyme A it is released here. So this GTP it will actually form ATP here. So this is actually the best example of substrate level phosphorylation. So actually from the substrate direct ATP synthesis occurs. So this GTP it will again convert it into the ATP by the action of enzyme nucleoside diphosphokinase enzyme. It will act on the GTP and will convert into the ATP. Now look at see here what happened to the succinate. So the oxidation of this succinate occurs. So mainly oxidation mean removal of hydrogen occurs. So this hydrogen it will remove here. Now this hydrogen it will gain by the FAD and will convert into the reduced form, that is FADH2. So simply up to this point 2 NADH2 it will form and 1 GTP it form here. So in this step the first FADH2 it will form during the conversion of succinate into the fumarates. So look at see here, this is actually the 4 carbon compound, this one and these are these 3 compound that are actually the 4 carbon compounds. Now here addition of water it will occur to the fumarate, and this fumarate it will convert it into the malate. So simply here the hydration of the fumarate occurs. So water molecule it will add it, and this addition of water molecule it will occur with the help of fumarase enzyme. So finally we will get the malate. So malate again it is actually a 4 carbon compounds. Now look at see here what happened to the malate. So simply malate, what happened to this malate? So again this malate it will oxidation of this malate it will occur, and finally we will get the 4 carbon compound that is called as oxaloacetates. So here the hydrogen it will remove, and this will gain by the NAD and will convert it into NADH plus H, it will form here. Now this enzyme it will this reaction it will mainly catalyzed by malate dehydrogenase enzyme. So the result we will get the 4 carbon compound that is oxaloacetate. So this oxaloacetate it will again combine with the acetyl coenzyme A and will continue the cycle. So this is the overall cycle, that is mainly the Palevink intermediate that are included in this Krebs cycle. As we look here, mainly 3 reactions that are irreversible in the Krebs reaction and in the Krebs cycles. Reaction number first, third and fourth, these reactions are irreversible, while the remaining reaction and the Krebs cycle that are actually the reversible. But instead of reversible reaction, these irreversible reaction that will go that will move in the upward direction because of the thermodynamic force. So these irreversible reaction that are have a strong tendency to move into the forward direction. Because of this it will create a thermodynamic pull, due to which the reversible reaction always move in the unidirection or in the forward direction. So this is actually the overall Krebs cycles. So finally, what is the importance of the TCA cycle or the function of tricarboxylic acid cycle? Now look at the first importance of the TCA cycle, that is the production of ATPs through this tricarboxylic acid cycle, as we have discussed in the energetic of the tricarboxylic acid cycle. So in each cycle of the TCA cycle we will get 10 ATP. So mainly this ATP it is utilized in various activity of the living organism. So mainly production of the ATP occur in this cycle. Now the second function of the TCA cycle, that is chlorophyll, is synthesized from succinyl coenzyme A. As we know that the succinyl coenzyme A, it is actually the one of the intermediate of the tricarboxylic acid cycle. Now this succinyl coenzyme A, it is used for the synthesis of the chlorophylls, and this succinyl coenzyme A, it is also used in the synthesis of the heme part of the hemoglobin. So this is actually the second importance of the TCA cycle. Now Krebs cycle is useful for the formation of proteins. So protein are actually the biomolecules. So the formation of this protein it is occur with the help of this tricarboxylic acid cycles. Now another importance, that is the intermediate of the tricarboxylic acid cycle, that is alpha ketoglutarate and oxaloacetate. Oxaloacetate are converted into glutamic acid and aspartic acids. So these intermediate of the TCA cycle, that is alpha ketoglutarate and oxaloacetate, that are converted into these amino acid, that is glutamic acid and aspartic acid. So now these amino acid it will form from the intermediate of the TCA cycle through transamination reaction. When transamination reaction of these alpha ketoglutarate and oxaloacetate occur, and it will form these amino acids. When the polymerization of this amino acid occur, mean this amino acid it will combine and it will form a large biomolecule, that is protein. So the result it will form proteins. Now the final function or importance of the TCA cycle, that is TCA cycle is useful for the conversion of fat into the carbohydrate through glycosylate cycle. So here Krebs cycle it is very important, because in this cycle the conversion of the fat into the carbohydrate through glycosylate cycle occurs. So this conversion it is occur with the help of these Krebs cycles. So this is all about the Krebs cycle. Thank you so much.